Swayam Prabha. Digital India. Educated India.
Welcome. So we have been discussing attention, and here our main objective is to look at network of neurons, how they change themselves from and when the animal or the organism is attending to a particular object to and when it is not attending to a particular object, and that change is being observed at a network level. And we found in our previous lecture that noise correlations, which is correlated variability in pairs of neurons, plays a big role in terms of how attention achieves enhancement in performance. So, in order to look at the mechanisms underlying the changes that we have talked about, we will be discussing a few experiments which show other than noise correlation what other aspects can play a role in attention. So, to begin with, it is known from many studies that nucleus basalis this is forebrain structure or basal forebrain that actually plays a role in modulating and providing cholinergic inputs to the entire brain. So, if we simply think of this as the brain from the basal forebrain region in the nucleus basalis, there are projections that go on to, this is just a cartoon, go on to variety, I mean in fact the entire cortical areas. So, this is nucleus basalis. So, acetylcholine is the neurotransmitter, and almost every region gets inputs in the cortex from the nucleus basalis. And we the first so the hypothesis is obviously that that nucleus basalis, can it somehow cause the changes in noise correlations that was observed in the previous experiment. So, this is, we are talking of a different group of people studying attention in a different way, and this is in the mouse where and looking at the primary visual cortex. So, what the experiment did was, first it is testing that if nucleus basalis is stimulated. So, it is known that there is activation of nucleus basalis in attention. So, the question is that if we stimulate nucleus basalis, then the effect on responses of neurons, does it match with attentional modulation or not? So, the experiment that was performed is that there is a stimulating electrode in the nucleus basalis. So, and in a mouse in the anesthetized state, a natural video is shown in front of the auditor, in front of the mouse, and responses from multiple neurons in V1 is recorded based and during that presentation of the video. And the data was collected on two cases, the same exact video. One is with NB stimulation electrical stimulation, and the other is without nucleus basalis stimulation. So, what the authors observed is that in the case of without stimulation and in the case of with stimulation, the biggest difference was not really in the rate responses. At rate response changes were not as large. The degree is small. The main observational changes were, if you looked at the responses simultaneously. So, let us say each row is one particular neuron's response, and we are recording simultaneously from n neurons. This is without stim, and in this case with stim. So, these are neuron 1 to neuron n. What was observed is that without stimulation, there was a strong correlation, that is between the neurons, that is all the neurons seem to fire in a synchronous manner, or rather many of them fired in a synchronous manner at the same time during features of the movie based on whatever receptive fields there are. That is neuron 1 responds here very little, and then when it responds, then neuron 2, neuron 3 and most of the other neurons also respond, and so on. So, that means that across neuron correlations, are high without stimulation, and with stimulation it was found that they became decorrelated. That is, these bands of activity at the same time by all the neurons together, that got washed out. So, if you think of high levels of activity in different neurons, they were not synchronized in time anymore. So, this is reduced dramatically, and basically it goes down to a very low and creates a decorrelation between neurons. So, if now you think about it, I mean in terms of what this can achieve in terms of behavior or in terms of information transfer or processing. So, you can see all the neurons behaving in the same manner simultaneously actually gives redundancy in the representation. That is, if all of them are saying the same thing, you might as well have only one or two of them say the same thing. The other, it was simply a waste of resources in terms of the amount of information to be transferred by those neurons. And if you have uncorrelated or decorrelated activity between neurons in response to the exact same stimuli, that would mean that the capacity of carrying information is higher now. So, the neurons are carrying separate aspects information of the stimulus, that is sort of the idea. So, that means essentially you are getting more resolution of whatever you are observing. I mean that is how you can think of it. So, this is one of the things that is observed with nucleus basalis stimulation, and the other large change that is observed is so this is also known as the that is reliability in responses. That is, if you look at the same neuron for the same movie on multiple trials, that is trial 1, trial 2 and up to trial k, let us say, without stimulation, and the same thing with stimulation. Then there is low reliability without stimulation when the movie is observed. That is, the neurons are not repeatable in their responses to the same stimulus. That is, each time there is a change in the pattern of responses to the same stimulus. So, this is what we are looking at is only one neuron, but multiple trials, and they are not correlated across trials. So, earlier we were looking at across neurons, and here we are looking at across trials, and this is actually low correlations in the without stimulation case, and in the with stimulation case they become more reliable in the sense they fire the same action potentials, they fire action potentials in the same time point based on the feature of the video that they might be encoding. And so, in every trial generally the spikes occur at the same point of the video for the same neuron, for one neuron. So, this reliability means that essentially an upstream neuron that is trying to decode based on this information has lesser noise in every trial in order to perform something based on the same inputs. So, that given these two effects of nucleus basalis stimulation, so reliability increase and decorrelation between neurons, the question then comes up is it the same as what is happening during attention? That is nucleus basalis the one that is actually performing this during attention or not? So, in order to test that, they changed the whole stimulation of nucleus basalis from electrical to optical. And they coupled it with behavior in a behaving animal. So, in an attentional task. So, first they replicated the, so what we mean by optical, if you recollect that now they are expressing channel rhodopsin, an opsin that is going to depolarize the neurons that are carrying it. They are and essentially make them more excitable or make them fire action potentials when when a particular wavelength light falls on them. So, they express this channel rhodopsin on the nucleus basalis cholinergic neurons, that is the neurons that have acetylcholine as their neurotransmitter. And so, as we know that the nucleus basalis projects all over, it also projects onto the V1 region, and so the opsin that is injected or that is expressed in the nucleus basalis, that is expressed in the terminals of those axons in the V1 region also. So, basically if you stimulate with that light, in this case a 473 nanometer light, these terminals get excited and they release the neurotransmitter based on what you know of synaptic transmission. Basically, they depolarize the synaptic terminals, calcium comes in, neurotransmitter is released, in this case acetylcholine. And so, they replicated that, okay, like electrical stimulation, with optical stimulation also they got the decorrelation between neurons while observing while the animal observed a movie, and similarly also there is increased reliability in responses, that is higher correlation from trial to trial. But to establish causation, one has to now couple this with behavior. So, there are two things that can be done. I mean, there is one more thing over this 473 nanometer case, that is, this is exciting or stimulating the nucleus basalis inputs in the V1 region. You can also turn off based on arc rhodopsin. So, you can express instead of channel rhodopsin that was expressed here, you can express arch instead, and with a that is a different opsin that hyperpolarizes the neurons, and this is stimulated by 590 or so nanometer of light. And so, this is suppressing or inhibiting the cholinergic neurons. So, the thing is that now if the nucleus basalis is actually doing this, and the changes that we see have a role to play in behavior, that is cross decorrelation of activity between neurons and increased reliability across trials, then if we turn off the nucleus basalis during an attentional task, we should get a reduced performance. Or if we provide more or excite the channel rhodopsin, excite the nucleus basalis inputs, we should be able to enhance performance. And side by side, we should also be seeing that the cross correlation across trials increases with excitation, and the cross correlation between trials reduces with inhibition, and cross neuron correlation is reduced with excitation, and cross correlation between neurons is not reduced when inhibiting the nucleus basalis inputs. So, indeed, so the in this case the animal was trained to perform this kind of a task, where in front of the animal. So, this is in the mouse, as I mentioned, and they are not very what should I say, very good viewers. They have poor visual acuity. So, they cannot resolve let us say oriented bars with very good resolution. So, an easy task was set up, and that is discriminating between vertical orientation bars or vertical gratings and horizontal gratings. So, either it was shown vertical gratings or it was shown horizontal gratings, and the animal had to discriminate between these two cases. It is an easy task. It is made difficult, slightly made difficult by changing the contrast between these dark and light regions. That is, you can have 100% contrast, that is based on the grayscale, basically the two limits. The white is at the one extreme and black is on the other extreme, and you can have gradually lower and lower contrast. And so, they did experiments where the animal had to detect the orientation changes. So, the performance of the animal is on this axis. So, with lower contrast, the animal had made errors, and with higher contrast their performance increased. So, this is the behavior of the mouse. So, this axis is contrast. So, essentially to make the long story short, with the nucleus basalis stimulation or with excitation with channel rhodopsin with the 473 nanometer light, they found improvement in performance. I mean up to the saturation level, when at 100% they were doing 100% correct. And so, this is with NB stim, optical stim in this case, and with arch, that is with NB inhibition, the performance was reduced. So, this is that case, and as we had also discussed, the reliability remained high in this reduced performance, and the across neuron reliability remained low, extremely reliability remained low, and across neuron correlations were high, that is higher redundancy. And obviously, the opposite was observed along with the improved performance, as we had seen with nucleus basalis stimulation earlier. So, with this with these studies, we can show how a network of neurons and the computation performed by those network of neurons to explain behavior requires incorporation of how they are behaving simultaneously, that is how their responses are related to each other during a particular task. So, the methods, the numerical or the quantitative methods that are associated with this, we had actually discussed earlier, that is noise correlations, and reliability is something that is very easy to compute, in the sense that you simply look at correlation of the entire spike train between trials and quantify it in terms of the correlation coefficient, and know what the reliability is. So, with this these studies, we can actually conclude our discussions on attention and how modulatory inputs like inputs from the nucleus basalis, in this case cholinergic inputs, play a role in providing a change to the entire network that goes beyond the enhancement in responses that we have been talking about in plasticity, that is the potentiation, depression, and so on. Of course, the long term potentiation, long term depression is at play here, but the bigger factor in this picture is how the whole set of neurons are changing together. And while some of the changes that we have talked about, for example, in the ferret case, if you remember that there is a change in the receptive field. So, let us say there is an enhancement in the side. So, let us say this is time, this is frequency, this is the STRF, and let us say this is the detected tone frequency. Then there is an enhancement from the passive case, which was let us say only an excitation band in this region, this particular frequency, which is its best frequency, that changes during behavior from the passive to the active case with this enhancement. And overall, when we look at the average and align them to the target frequency or the detected frequency, this is the target frequency. Now, we are combining these results, the difference between the two, and averaging across many trials, many neurons, we see a large enhancement and maybe some depression on the sides. And so, although during behavior the change that is occurring and the average change that is occurring is an enhancement that we are looking at, many of these neurons switch back to their original receptive field, that is, or rather from this change state they go back to their original receptive fields. However, many neurons actually continue to be in this change state, which is actually a long term change now. So, however, in the case of attention, the kind of changes that we are talking about, or at least the examples that we are talking about, it has not been really studied that whether there are some permanent changes that are induced that carry on forward beyond that task. So, that I mean, most likely we can hypothesize that some of them do, just like in this particular case, some of them continue to have a changed receptive field while some switch back. That part remains to be seen, what actually happens. So, with this we come to the end of our discussions on attention, and next we will be discussing more on incorporation and the implications of implementing long term plasticity in some phenomena, in particularly those during development. Thank you.